All right, so I'm going to just go over some of the background information about DNA and RNA that might be helpful in understanding how it works in viruses. So DNA is made up of this phosphate sugar backbone, so that's kind of represented here. And then we have these bases that are nucleotides. So we always have C and G pairing up and T and A pairing up. And within kind of the DNA double helix that we're all familiar with, that's actually two strands of DNA. And so each of these sugar phosphate backbones, and then they're connected through these nucleotide bonds. Mm. All right, so each strand has directionality, and you can see here we have a 5' and a 3' end, and on the kind of across on that second strand, it's opposite, 3 to 5' prime. And so those numbers come from the structure of the sugar in the backbone where the phosphate's going to be attached. So you can see this pentagon shape is similar here, and on this side, this is the 3 uh, carbon, I guess, and at the 5, it's at this end. Um, And so you don't need to worry about the chemistry, but what's important here is that the direction matters, the directionality of these strands. So they're not kind of identical strands. They're complementary to each other and they're anti-parallel. They're going in kind of opposite directions. Um, all right. And when I say that they're complementary, it's that these base pairs are complementary. So A is always going to pair with a T, T is always going to pair with an A, and then we have G paired with a C, C paired with a G. And so this complementary base pairing on anti-parallel strands is really kind of foundational for how DNA gets copied and how genetic information ends up being kind of read later on to turn it into proteins. And so with DNA replication, if we look at this DNA ladder, when new DNA is made, it's done in what's called a semi-conservative way, a semi-conservative model. And so the two strands will separate from each other, and then new DNA is built on top of that. And so if we kind of zoom out, what you end up with is the the original strand is breaking apart, and as those split, new DNA is built on top of that. And so at the end, you end up with two identical molecules of DNA, identical to each other, and each is half new and half old DNA. And the enzyme that actually kind of does the building of the new strand is will be a DNA polymerase. There's different types of DNA polymerase, but for our purposes, just knowing that DNA polymerase is what is actually going to kind of bring those nucleotides in and the phosphate sugar backbone and build that new strand. And so it'll move along the original strand, add the nucleotides, following those base pairing rules, right? A always with C, G with C. And so that sequence of the old strand that acts as a template to what the new strand is going to consist of, what the order of the code is going to be. And so this always is going to happen in one direction. So the DNA polymerase can only add new nucleotides to the 3' end of a new strand. And so it's always working, building from 5 to the 3 end. And this ends up being really important when we start looking and thinking about how viruses copy their genomes inside their host cells. And this is just a little bit of a summary of what the key info we need to know to understand virus replication. And so there always has to be a template strand. The DNA polymerase builds that complementary strand. It's always in the 5-3 direction. And here is kind of some of that going on. So we have DNA polymerases here. They're building. Here's kind of a simpler one to see in the 5-3 direction along that leading strand. Something similar is happening on the lagging strand. All right. And so far, that's just about copying DNA, but copying and actually expressing DNA is a little bit different. So a gene is going to be a specific stretch of DNA that contains instructions for making a protein. And so to use that information, the cell first makes an RNA copy of the gene, and this was called a messenger RNA or mRNA. The mRNA then serves as a template for building a protein. And so it goes from DNA, there's transcription is the kind of process that occurs to end up with mRNA. And then that is what's referred to as translated. That's the process there into a protein. And so when this happens, we have our two strands, our double-stranded DNA. One of them is considered the template strand. So that's the 3-5. And so that's the one where that ends up being kind of transcribed. So the mRNA that's built on here is built from the 5-3 direction, and it'll be complementary to the template strand. It's going to be basically identical to that non-template or coding strand. But we do see a difference because it's RNA versus or RNA versus DNA. The G's, any G's, C's, and A's are going to be the same, but anywhere there's a T on the D, anywhere there would be a T on that coding strand, it's going to end up being a U on the messenger RNA. All right, and so during transcription, an enzyme called RNA polymerase. So this is very similar to DNA polymerase and the function of DNA polymerase, except it's building RNA polymerase. And what you have in animal cells is DNA-dependent RNA polymerase. So it depends on a template of DNA to build RNA molecules. And so it will make kind of the complementary strand and build that. As I said, the U's replace the T's, so it's uracil instead of the thymine. It's going to build in the 5' to 3' direction. So that directionality matters, and it's working in the same direction. It's always kind of being built in that way. All right, so then at the end of transcription, the DNA will just stay in the nucleus where it was. The mRNA carries information needed to make a protein. And so that mRNA is now used as a kind of a template, like we see here, to build an amino acid chain. During transcription, only one strand of the DNA is used. I went over that a little bit, but there's, to make it all more confusing, there's a bunch of different names that are used for these things. But basically the DNA, the template strand, is the one that goes from 3' to 5'. The mRNA is complementary to that, and it's built using that template strand as a template. It's sometimes also referred to as an antisense, a negative, or a non-coding strand. mRNA ends up being essentially the same as the non-template strand, also called the sense, positive, or coding strand of DNA, except those T's are replaced by U's. mRNA is the 5 to 3 direction. All right, so these kind of basic rules for the genetic codes, templates, polymerase, and building 5 to 3, we can think about kind of what's key for viruses. To copy a genome, there needs to be an appropriate template and the appropriate polymerase. To make proteins, for translation to occur, there has to be some RNA that goes from the 5 to 3 direction, or it can be called sense or positive RNA, and that's the same as the mRNA.